There we go. Okay. So again, just to reiterate, functionality of the kidneys, right? The major goal is for excretion of waste from our body, or excretion of waste products. So anytime we're taking stuff in through the intestinal tract, we're not going to, 100% use everything to maximum efficiency. We do have waste, and part of that solid waste is evacuated through us in the form of a bowel movement and fecal matter, and then the other part of that is liquid waste that's gotten rid of us in the form of urine, right? So not only do the kidneys serve to get rid of liquid waste products and generate a form of liquid waste, they also regulate fluid control, or they control fluid balance within our body. So they regulate the fluid content of our body. So more specifically, the blood volume that we have, so the amount of blood that's passing through our arteries and our veins, the volume of that. Remember, it's not just a bunch of red blood cells. It's actually, a significant portion of that is plasma. And so, controlling the water content of that is a job for the kidneys as well. We're also going to control what's inside of that liquid secretion. So we're going to control the kinds of solutes that are dissolved in there. We're going to control how much sodium, chloride, potassium, calcium, all of these, important trace minerals are controlled in their amounts by the kidneys, and how much of that we have inside of our inside of our circulatory system. Okay. Very importantly, they are going to help control fluid pH. So they're going to balance the amount of hydrogen ions that are floating around within our bloodstream. So we have an important regulatory mechanism that controls blood pH that the kidneys are responsible for. And what's kind of interesting is it's actually linked to the lungs as well. So the lungs have a role in that too. We also control the amount of red blood cells that we have. So erythrocytes are controlled by the hormone that the kidneys themselves produce called erythropoietin. And so that erythropoietin will stimulate stem cells in red bone marrow to upregulate the production of red blood cells overall. And since the kidneys control all different kinds of ions that are present within, the dissolved solutes of our blood, since they control things like calcium as well, calcium is an important precursor for the synthesis of vitamin D and vitamin A, and kidneys play a role in controlling how much vitamin D we synthesize as well. Okay. So kidneys do a lot of different things, and one of the major things that we're going to talk about today is some of the excretory functionality, okay? How all these tubes are linked up with one another. Okay, so again, just to reiterate the position of these organs within our body, here we've got kind of a faded view of the abdomen here. And we've got our liver that sits up here. We're missing an important muscle organ that's covering all of this called the diaphragm, which kind of, separates the abdomen from the chest cavity. But sitting just underneath and behind the liver, we've got our kidneys here, okay? And their vascular supply comes from a paired set of arteries and veins that are coming off and going to the aorta and the inferior vena cava, respectively, called the renal vessels. So the renal arteries and renal veins. So the key word for today referring to the kidneys is renal. So I'll type that in our group chat as well. So renal refers to anything kidney related, okay? And then anything that's like adrenal is going to refer to the adrenal glands that sit on top of the kidneys here. Okay, so we've got our renal veins, and then the renal arteries sit underneath them. Now, what's kind of interesting too is, and again, this doesn't happen too often. Usually, the arteries are actually going to be more superficial relative to the veins, but in this case, the renal veins, are actually more superficial, relative to the renal arteries. So when you dissect a kidney, the major vessel that you see here that's on top is actually the vein, and the artery sits just underneath it. Okay, so pretty much like the way that I think about kidneys, and a way that you can think about them, is kidneys are just kind of these like hyper specialized trash chutes, right? So all of the liquid waste gets thrown into these things from our bloodstream, and then it's just a bunch of tubes that are all connected to one another that eventually empty into bigger and bigger tubes until finally you get to the biggest tube of waste direction, which is going to be the ureter. So the ureter is the one that's going to connect the kidneys to the bladder. Okay, so ureter. So the ureter goes from the kidney to the bladder. Okay, and then the urethra is from the bladder to the external environment. So just an important distinction there, because that's like whenever I ask students questions, regarding the urethra or the ureters, invariably, they get those two things mixed up, whether it's simple identification or some of the physiology associated with them. The ureters and the urethras, tend to get mixed up. So ureters from kidney to bladder, urethra from the bladder to the external environment. Okay. So these ureters here are important because they actually have some smooth muscle and they pass liquid from the kidneys into the bladder via peristaltic motion. So very similar to the small intestines, the duodenum, large intestines, how we move things through our intestinal system. We do the same thing with moving fluid from the kidneys down to the bladder. Okay, and so then the bladder is going to play an important role for storing urine until it reaches a certain size. Then we have something called a micturition reflex, which is going to trigger a sensation for us to need to pee. Okay. And so you can see some of the other associated vasculature of these areas as well, but just note the position of the urethra here. See, and I just did it. The ureter, note the position of the ureter, it's actually going to be more superficial relative to the rest of the vasculature in the abdominal cavity here. Okay. One of the exceptions is going to be if we refer to kind of male excretory anatomy versus female excretory anatomy. Males have these other tubes that are going to come up and loop over the ureters, and we call those the vas deferens or the ductus deferens. So those are the tubes that are linked to the testicles and carry seminal fluid, or actually at that point carry sperm. Okay, so this is just the position of the vasculature and the structures of the kidneys relative to everything else in our body. But remember, another interesting thing about the kidneys, and this also corresponds to the genitals of the body as well, whether they are ovaries or testicles. They're what we call retroperitoneal structures, right? So remember, they sit behind the parietal peritoneum of the body, of the inside of the body. Remember, so this connective tissue that isolates the body wall with like our skin and musculature and everything from the inside of the body cavity here is protected by this layer of connective tissue called the peritoneum. Okay, and this parietal peritoneum divides the abdominal cavity into different sections here. And the kidneys themselves, along with all the other associated vasculature and parts of the intestines as well, are going to be behind that. So they actually sit behind this connective tissue. So what does that mean? Why is that significant? Well, what that means is that when you look at these things in vivo or in an actual specimen, they tend to look like tumors at first because they're these big masses that are growing behind the body wall, and it can kind of look abnormal because all the other guts are actually sitting inside the body cavity. But the kidneys themselves form behind this. And there's embryological significance for that as well, and part of that is for protection. Part of the development of the kidneys here is closely linked to the development of things like the ovaries and the testicles as well. So that's why it's usually when we're referring to it in embryological terms, we call these things the urogenital system because they develop very closely with one another. The vasculature that's associated with them is very, very similar too. But so the kidneys and their associated vasculature are retroperitoneal. Okay. So they sit behind the body wall. All right. And so, again, they're going to have their own sets of connective tissue as well. So they've got the renal fascia here, the renal capsule here as well. That's going to kind of contain the actual organ, or create what we view as the functional organ of the kidney. And then the kidneys themselves also have a renal hilus here as well, where all the vasculature is going to be entering and exiting the kidneys too. Okay. So just note kind of the position of that here in our view. We're looking, from the feet towards the head. And we can tell that because obviously it tells us that it's an inferior view. We know that this is the anterior posterior side, and then we can see the left lobe of the liver that's positioned here, okay, and then our peritoneal cavity. Okay, so here we've got a frontal section through the kidney, right? So this is a classic section that we do in dissection where you just literally cut the thing in half, right? And so we like this view because it gives us the best view of not only the vasculature, but also the open channels that we have, where all of these, we're gonna learn about this in a second, these collecting ducts that form the structure called a renal pyramid, which is basically all of our liquid collecting tubes empty into these things, okay? And we call these calyces, or singular would be calyx, right? So the calyx here, what we call a minor calyx, is going to contribute into a bigger area over here called a major calyx, which contributes over here into the renal pelvis and then ultimately the ureter here. But we're going to focus a lot on the functional units of the kidney today and kind of how they operate. And so we can see that, like most of the things in our body, they have a rich network of arteries and veins, right? And that's especially important for these structures because they're going to be controlling a lot of things that are present in the blood, dissolved solutes, blood pressure, blood volume, right? All of these things are going to be controlled in part by, what the kidney does. So it makes sense that we're going to have a rich network of all these little capillary beds here. There's two major parts of the kidney, what we call the medulla, which is going to be this area where we have these renal pyramids, and then this outer layer that's going to be fainter in color, not the renal capsule, but kind of just underneath it. We call that the cortex. So the cortex is the outer layer, the medulla is going to be this kind of inner layer, right? So you can kind of draw an imaginary line and where the top of these renal pyramids are, and that's going to be, everything in here is going to be our medulla, and then the kind of outer layer that's beyond the medulla is what we call the cortex. Okay, and so we've got different kinds of nephrons that sit within both the cortex and the medulla. And again, just that term hilum referring to that indentation in the organ here. Okay. And here's what that looks like in vivo. So this is an actual human kidney that's been, sectioned in the same exact way. And we can see we've got our medular region here with the cortex out here, but each one of these dark spots is a renal pyramid. And that renal pyramid is literally just a collection of what we call collecting tubes or collecting ducts. They're going to provide the filtrated urine into these large cavities. Okay. Oh yeah, no worries. Thanks, Cynthia. Okay, so here's kind of like our two functional areas broken down a little bit more. So the functional unit of our kidney, so the actual thing that's doing most of the filtrating and reabsorbing water or getting rid of water if we don't need it, or getting rid of solutes or maintaining solutes if we need it, is what we call the nephron. Okay. So the nephron is the functional unit of the kidney. And so we're going to look at that in a little bit more detail here in a sec. And the nephron is going to consist of several different structures, but really in total, we can kind of summarize that with this image up here on the kind of where I'm circling. So we've got a little structure that we call the renal corpuscle, which is made of two different things. So that's how you say that term, corpuscle. It's made of a rich network of arteries that we call the glomerulus, right? So that's how you pronounce that, glomerulus. And then the actual kind of open vasculature that's not associated with the circulatory system, but it's going to be associated with the filtrating part, which is what we call the Bowman's capsule. That Bowman's capsule is going to receive all the filtrate from the bloodstream, and it does that through things like osmotic pressure and blood pressure. And then all of the stuff that we're trying to get out of our bloodstream, whether that's water, proteins, ions, whatever it is, all gets put into this tubular system, or what we call the convoluted tubule system, right? And there's three major parts that we'll address here in a sec. But the first part is this little close squiggly bit that we call the proximal convoluted tubule. So most of our water retention occurs here as well. We retain up to like 65% of the water from our diet within the proximal convoluted tubule. But then we're also going to control a lot of ion concentration in our bloodstream in this next bit that we call the loop of Henle. Right? Now, ion concentration is important because it's going to determine a lot of where water and other kinds of fluid are going to be placed within the body. Are we going to get rid of these things? Are we going to excrete them and reabsorb them into the bloodstream? Or are we going to maintain them within the convoluted tubule system and get rid of that stuff? The loop of Henle here controls a lot of where that stuff goes. Okay, so the fluid will move through the loop of Henle here. And we'll talk a little bit more about the loop of Henle. There's some cool animals that have some interesting takes on the loop of Henle that help them survive in hot, arid climates. And then we move into the distal convoluted tubule, where we're going to do a lot more water reabsorption as well if we need it. And then as we move from the distal convoluted tubule to the next bit, this is what we call a collecting duct. All right, so the collecting ducts of a whole ton of them, all bunched up together, are what make up the renal pyramid. Okay, so the renal pyramid is just a collection of collecting ducts all smushed together, right? And so we've got two different kinds of nephrons. We have cortex-based nephrons and then medullary-based nephrons. And so the distinction there is, how much of the loop of Henle is present within the medulla or within the cortex. If it's mostly within the cortex, it's a cortex nephron. But if it's mostly in the medulla, it's what we call a medullary nephron, okay? Or what we call a juxtamedullary. So there's this other kind of fancy word. This is like your $10 word for the day. It's called juxtaposition. All right, so this is a fun little word to throw out there, but all it means is to be kind of transposed upon or adjacent to in position. Right, so you'll see this come up a lot in some of the terminology associated with the kidneys. And really, for the context that we're focusing on with it here, it just means a structure that is next to something else. So if it's a juxtaglomerular cell, it's going to be a cell that's right next to the glomerulus. If we've got a juxtamedullary nephron, okay, this nephron is going to be next to the medulla, right? So juxtaposition is to transpose position. So you can think of like if you are at point A and somebody else is at point B, and if you guys switch places, that would be a juxtaposition, right? So what we're referring to here is close proximity to something else, right? Okay, so what's also not pictured here is each of these collecting ducts, the loop of Henle, the proximal and distal convoluted tubules, all have a ton of arteries and veins associated with them, tons of capillary beds that are wrapped all around them, because what we'll find out is that a lot of the time we retain a lot of the water that ends up getting into the kidneys, right? Because most of our body is composed of water, and water is a super important portion of our diet, right? This is why you can't go for long periods of time dehydrating yourself without diet. But on the opposite hand, you can starve for up to 2 weeks and you can still live, right? But water, you need a constant supply of water in order to be able to survive. Okay. So here's kind of the functional unit of our kidneys, our nephron. We're going to break some of this down here. So this first bit, this is our renal corpuscle. So we've got a little capillary bed that comes in here. So we've got kind of two ends of our capillary bed. One part that's called the afferent arteriole. So this is the one that's going away. So afferent means away. Okay, and then afferent is towards something, not away. Efferent is away. So the afferent arteriole is bringing blood towards the renal corpuscle right here. Okay, and then the efferent arteriole is taking blood that has gotten rid of filtrate away from the renal corpuscle and back out into the circulatory system. All right. So again, the renal corpuscle composed of two major things: the glomerulus, which is just the capillary bed that's composed of the afferent and efferent arterioles, and then the Bowman's capsule, which is the filtrate tube that surrounds it and essentially grafts onto the actual cells of the capillary bed. So this Bowman's capsule has a visceral layer that sticks directly onto the capillary bed, and it's got a parietal layer that kind of makes up this outer bit right here. And so we can see it's got this large opening that's going to be able to receive a lot of filtrate from the capillary bed here and push things into the proximal convoluted tubule. Okay, so the renal corpuscle consists of the Bowman's capsule and the glomerulus. Bowman's capsule is the enlarged end of an nephron, which is indented to form a double-walled chamber, right? So each of these walls is going to serve an important function. So the Bowman's capsule surrounds the glomerulus, which is a network of capillaries, and blood flows from the afferent towards this bed, and then the efferent brings blood away. All right. So then, kind of looking at this a little bit further, we can see we've got some simple squamous epithelial cells that make up the outer lining of our Bowman's capsule here. But then what's kind of hard to envision or picture here is all of this kind of purple interweaving and overlapping stuff right here. Those are all cells that comprise the visceral layer of our Bowman's capsule. Okay, so the Bowman's capsule essentially sticks right onto the afferent arteriole here and is going to be able to leach all of the nutrients, or I should say not all the nutrients, all the waste products that our body doesn't need from the circulatory system, whether that's water, whether that's ions, whether that's salts, whether it's other kinds of waste, right? So all of those things are going to move from the arterial bed here into the renal corpuscle, from the glomerulus into the renal or into the Bowman's capsule here. Okay, so then once fluid gets into here, it goes out through the proximal convoluted tubule. All right, so the visceral layer of the Bowman's capsule covers the glomerular capillaries. Fluid from the blood enters the Bowman's capsule by passing through the capillary walls and the visceral layer of the Bowman's capsule, namely through hydrostatic pressure, so the amount of fluid being forced against the arterial walls, but also osmotic pressure, so concentration gradients are going to be controlling the movement of solutes into the Bowman's capsule. Now, if you had to guess, if you're trying to figure out, okay, is the concentration of salts going to be greater inside the corpuscle, or is it going to be greater? Is it going to be greater inside the glomerulus, or is it going to be greater inside the Bowman's capsule? And it's going to be greater inside the capillary bed because that's where all of the waste is initially, and then it all moves out from there. But if in terms of the active gradient, we're going to have a higher concentration of all of our waste products inside of the capillary bed here compared to inside the Bowman's capsule. So again, that's one of the controlling factors that moves things from our bloodstream into the actual waste processing part of this. So from there, fluid passes into the proximal convoluted tubule of the nephron. The juxtaglomerular apparatus consists of cells from the wall of the afferent arteriole and the distal convoluted tubule. Okay, so this juxtaglomerular apparatus is important because this is going to secrete different kinds of hormones that let the brain know, okay, is our blood pressure low or high, right? And so this is going to control things like our need or our sensation of thirst or the need to drink more water, or if we need to trigger a micturition response where we need to produce more urine, right? Where we need to dilute our urine, I should say. So each of these cells, so the macula densa and the juxtaglomerular cells, sense pressure changes in the arterial wall. So if this wall is nice and full, blood pressure is high, that means we don't really need to drink a lot of water. But if blood pressure is low, they're going to secrete hormones that tell the brain, hey, we need to be drinking more water here. Okay, so that's the significance of the juxtaglomerular apparatus. These cells that are adjacent to the glomerulus here are going to assist with recognizing blood pressure changes. Okay. Okay, so then another close-up look at the capillary bed with the visceral Bowman's capsule layer. We can see that the arterial walls of this capillary bed are fenestrated. They have holes in them, right? So each of these fenestrae are going to contribute to the ability of large solutes moving out of the capillary beds without really needing a ton of energy to do so. So this first process is fairly unselective. Basically, if something's big enough to fit through those holes and it's moving down its concentration gradient, it's going to do so. Okay. So the glomerulus is composed of fenestrated capillaries. So they've got little holes in there. The visceral layer of the Bowman capsule consists of specialized cells called podocytes. And spaces between the podocyte cells are called filtration slits. So that's where everything ends up filtering out. Okay. So the filtration membrane consists of the fenestrated glomerular capillary endothelium, a basement membrane, and the podocyte cell processes. Fluid passes from the capillary through the filtration membrane into the Bowman's capsule. Okay. So each of these fenestrae are going to allow for solutes and other kinds of waste products to passively move from the bloodstream into the Bowman's capsule for filtration. Okay, so a note on vasculature here, all right? So as I mentioned, there's a rich network of vasculature associated with the functional units of the kidney. So the kidney itself and also the nephron, right? So if we look at our kidney over here, if we start with the renal artery, so if we're a blood cell going into the kidney, we would first start out with the renal artery and then move to the interlobar artery, so in between the lobes of the pyramids here, okay? And then into an arcuate artery, which moves closer towards the cortex up through the medulla, and then again through another interlobular artery, right? And then moving from there, all right, we would go into the afferent arterial, right? So the afferent arterial, glomerulus, efferent arterial, right? And then out through the peritubular capillaries, so all of the vasculature that's going to be associated with the collecting ducts. So this is important because this is where a lot of our water and other kinds of solute retention are going to occur. So we need a rich network of vasculature there, all right? So then we come back through the vasa recta, which is just a fancy word for capillary bed associated with the loop of Henle here, and then into the interlobular vein. So interlobular vein, arcuate vein, interlobar vein, and then finally through the renal vein. Okay, so once again, we start out with the renal arteries, we get smaller and smaller, interlobar, arcuate, interlobular, and then into the actual nephron right here, afferent, glomerulus, efferent, peritubular capillaries, so surrounding all of the areas where we're going to be secreting or we're going to be trying to get rid of water, but if we need to retain water, we can pick it up here, and then everything can get back into the circulatory system through the vasa recta and the arcuate veins, and then ultimately going down to the renal veins here. Okay, so that would be the trajectory of a red blood cell moving through the kidneys. Okay, so filtration pressure is going to be very, very important for producing urine, right? So we can kind of break up urine formation into three separate parts. We have filtration, tubular reabsorption, and then finally tubular secretion. So with filtration occurring, it's the movement of materials across the filtration membrane into the Bowman's capsule to form filtrate. Now, as I mentioned, this process is not selective. It's not going to be really caring about what's getting in here. If it's big enough to get out of those fenestrae and through the podocytes of the visceral Bowman's capsule, it's going to do so, right? So the filtration part of this is really just controlled by blood pressure and osmotic potential. If we have enough force behind us, we'll move, and if we're big enough to fit through the hole, we're going to do so, okay? But then sometimes, that's not always a good thing, right? Sometimes we need to be able to control what we're getting rid of. Sometimes we need to maybe reabsorb some extra sodium or chlorine or potassium or or fluoride or any kind of thing, any kind of other trace element that we may have in our excretory system. So I just want to make sure my coffee here doesn't get too diluted. All right. So the tubular reabsorption part is going to control how diluted our urine is. Are we going to need to maintain or recuperate any kind of water that's gotten into the Bowman's capsule during the filtration process. So solutes are reabsorbed, okay, through this purple arrow, so salts and other kinds of ions, across the wall of the nephron into the interstitial fluid by transport processes. So this part requires energy, right? So we have specialized cells that are going to be moving solutes out of the nephron if we need them, okay? Water also gets reabsorbed by osmosis. So this is passive. This doesn't require energy. So if we want to reabsorb any kind of salts, sodium chloride, potassium, anything like that, we need to expend energy to do so. But water is nice because it'll just move down its concentration gradient. It's relatively small polar molecule, so it can move through some special kinds of transport proteins pretty easily. So water gets reabsorbed by osmosis. Water and solute pass from the interstitial fluids into the peritubular capillaries, right? So the tubular reabsorption part is important because this is where we retain water as we drink it, right? So this is going to be where that occurs. And then we have our actual tubular secretion where we produce kind of like our functional urine component, where any solutes or water that is not needed end up making their way back into the collecting ducts of the nephron and end up getting into the ureters and then ultimately the bladder and then the urethra. Okay, so solutes end up getting secreted across the wall of the nephron into the filtrate, right? So sometimes if we've got too much solute within the bloodstream, we can force that stuff back across right here, right? And remember, we've got some important chemoreceptors that are going to be able to determine that. So we have other kinds of proteins and other kinds of solute recognizing proteins that are present in the bloodstream here that are going to say, hey, okay, we actually don't need a lot of these solutes, so you get back over here. You can get it back into the filtrate, right? We don't need this. We're going to secrete that as urine. Now, funny thing enough, most of our urine is actually water, right? Even at our most dehydrated, urine is usually only at most 10% urea, okay? Which is why, if any of you ever watched like Bear Grylls or anything like that, insert meme of Bear Grylls drinking his own piss, right? He's able to drink his own urine in survival situations a lot of the time because it's mostly, even in super dehydrated situations, it's mostly water. The urea, obviously, I would imagine doesn't taste great, but it's still mostly water, and that's water that he can put back into his system. But if we're very, very hydrated, urea content is usually only around like 2 to 1% at most, right? So most of the urine that we excrete is water, even when we're very, very dehydrated. But we can control that with things like filtration pressure and active and passive transport that's going to occur within places like the proximal convoluted tubule and the loop of Henle. Okay, so what's kind of cool is, as I mentioned before, quench your thirst, right, Hugo? Insert all of your fun Bear Grylls memes right there. So with filtration pressure, right? So pressure gradients are going to be very important, just like with any kind of other capillary pressure. That's no different here. So filtration pressure takes into account things like glomerular capillary pressure, which is usually quite large. It's large enough to outweigh the capsular pressure of the actual Bowman's capsule here, along with the colloid osmotic pressure. So the osmotic gradient that's going to want to force solutes back into the arterial flow because the gradients are being set up that way. But the nice thing is when everything's functioning correctly, that glomerular capillary pressure is large enough to force everything against those concentration gradients. So glomerular capillary pressure, the blood pressure within the glomerulus, moves fluid from the blood into the Bowman's capsule. Capsular pressure, the pressure inside the Bowman's capsule, moves fluid from the capsule into the blood. Colloid osmotic pressure produced by the concentration of blood proteins moves fluid from the Bowman's capsule into the blood via osmosis, right? Filtration pressure is equal to the difference between each of these things, all right? So the greatest pressure here is the glomerular capillary pressure because that's still going to be responding to pressure from our heart. So as our heart is boom, 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 forcing blood throughout our body, even down at this little capillary level, it still has a large enough pressure gradient to outweigh things like capsular and colloid osmotic pressure, okay? But again, the nice thing about each of these other pressures being valued here, being significant, is that even at the glomerular level, we can still retain water and blood proteins if we need them, if for some reason they get across those fenestrated capillaries right here. Okay, so let's kind of break this down here. So on the left-hand side of this figure tells us a lot of cool information. So we can see on the left-hand side that we have the relative concentration of interstitial fluid in what we call milliosmoles, right? So it's kind of an arbitrary unit, don't really worry about it, but obviously the bigger the number, the more concentrated that interstitial fluid is, meaning that there's less water in that area, right? So the lower the number, there's more water there, right? So approximately 180 liters of filtrate enter the nephrons each day. 180 liters, right? Of that volume, 65% is reabsorbed into the proximal convoluted tubule, okay? So you get 65% of that water back via the proximal convoluted tubule. In the proximal convoluted tubule, solute molecules move by active transport and co-transport from the lumen of the tubule into the interstitial fluid. Water moves via osmosis because the cells of the tubule wall are permeable to water, right? So you'll find that water actually gets to kind of cheat a lot of the energy requirements of this stuff, but the solutes and other kinds of charged particles don't have that luxury, right? So here in the loop of Henle, right, approximately 15% of the filtrate volume is reabsorbed into this segment of the descending limb of the loop of Henle. The descending limb passes through the concentrated interstitial fluid of medulla, so super, super high concentration. And again, there's going to be less water here, so it makes sense we're going to be able to retain a little bit more water at this point, right? Water moves by osmosis from the tubule into the more concentrated interstitial fluid. By the time the filtrate reaches the tip of the renal pyramid, the concentration of the filtrate is equal to the concentration of the interstitial fluid. So down here, everything's at equilibrium. There's no net movement of fluid, of water or solutes. But once we reach the ascending limb over here, this is where we're going to have a lot of our ion control. So things like salts, sodium chloride, sodium ions, chloride ions, potassium, all those kinds of things are going to be controlled with the ascending loop of Henle. So the ascending limb of the loop of Henle isn't permeable to water. Solutes are allowed to diffuse out of the thin segment, whereas sodium gets actively transported and potassium and calcium are co-transported out of the thick filament right here. So the thin and thick portions of the loop of Henle are nice because it allows for a distinction between what kinds of things can pass across this area. In the thin segment, salts can passively move across, but if the filtrate reaches our thick limb segment, then things require energy or they need to be transported along with something else in the form of a co-transport. Okay. So once we reach we're kind of nearing our distal convoluted tubule over here, the volume of the filtrate doesn't change as it passes through the ascending limb, but the concentration is greatly reduced. Okay, by the time the filtrate reaches the cortex of the kidney, the concentration is approximately 100 milliosmoles, which is less concentrated than the interstitial fluid of the cortex, right? So it's fairly diluted. We're going to be, forcing a lot of water into our tubular system here. Okay. Then finally, the distal convoluted tubule, along with the collecting ducts, are going to help us reabsorb, more water from the excretory system here. So these are permeable to water if antidiuretic hormone is present, right? So if that part of our hypothalamus is releasing this antidiuretic hormone, saying like, hey, we don't actually, need to urinate, we're actually kind of dehydrated, we want to retain more water, this area becomes permeable to water, allowing us to retain more water, right? So if ADH is present, water moves by osmosis from the less concentrated filtrate into the more concentrated interstitial fluid, right? So this is important setting up that gradient here because we allow for water to passively move from the waste system back into an area where we may need it. By the time the filtrate reaches the tip of the renal pyramid, an additional 19% of the filtrate is reabsorbed. 1% or less remains as urine when ADH is present. So if antidiuretic hormone is present, we want to retain as much water as possible. So 1% remains as urine once we reach the end of our journey right over here. Okay. So this is kind of the journey of fluid through our nephron. This entire image is a really good representation of the actual nephron. So we've got our renal corpuscle with the glomerulus and the Bowman's capsule, proximal convoluted tubule, loop of Henle, and then the distal convoluted tubule, and then our collecting duct over here. So what's kind of cool about animals that are desert adapted or adapted for areas that don't get a lot of rainfall is they have these really, really long loops of Henle that allow them to spend a lot of time controlling where their solute concentration moves. And in doing so, they're able to essentially survive off of most of the water that they just get from eating different kinds of fruits and nuts off of little woody bushes or things that you could find in desert climates, or other kinds of fruits that they may find on things like prickly pear cactus and that sort of thing. Really good example of this is the kangaroo rat. So if you check out, or you can go on Google, look up kangaroo rat. They're a protected animal species, especially in Southern California, but they're native to this area. And there's quite a few of them out in Riverside, kind of back behind Lake Matthews. One of my buddies in undergrad, he and his parents lived out there, and so when I would go and visit him, we would see them kind of like hopping along the hills. But kangaroo rats are kind of adorable. They look like these little furry T-Rexes, but they're really, really interesting for a host of other, anatomical reasons. But a really cool physiological adaptation that they have for their environment is based around their ability to survive off of, or getting all the water from their diet that they need just from fruits and nuts and that sort of thing. And they virtually rarely will drink water. And you'll find this is the case for a lot of other desert adapted animals. Cats, not necessarily domesticated cats, but wild cats, like big cats, jaguars, lions, tigers, those sorts of things. Cheetahs are all adapted to have really well developed loops of Henle for maximum water control or maximum solute control about where water is being positioned in their body, right? Because if you can control the solute position, you can create hyper concentrated areas of the kidney with the inner medulla and the outer medulla along the cortex, which allows for you to retain more water as you need it, right? Because if the inside of the collecting duct is going to be more dilute, that means there's more water there. And if the outside of it is more concentrated, water wants to be over there, right? Water doesn't want to be where a bunch of water is. It wants to be where all the extra solutes are. So water will move from the waste processing system back into the body so that we can retain more water, right? Yeah, so I mean, and even still, like domesticated cats, you'll find that most of them don't drink like a ton of water, but they'll kind of drink sporadically here and there. But they will drink far, far less than something like a dog, right? There's different kinds of dog species that are adapted to desert climates, but the physiology of animals that live in extreme environments like the desert is a really, really interesting thing to study. And just a quick side note with this whole thing is a side note, but also with kangaroo rats, their muscle physiology is really, really interesting because we call them kangaroo rats because they look like regular sized kangaroos, right? But actually kangaroo rats in terms of the biomechanics are completely different than their actual kangaroo cousins. And what's kind of interesting is that kind of holds up as you scale animals down. So once you get kind of below, I think it's about 500 grams, biomechanics that are kind of traditional and what we would assume other animals can operate around become very, very different. And by that I mean kangaroos can kind of hop around for free. They don't have to expend a ton of energy like forcing themselves to hop. Once they're hopping, their tendons are really stretchy, and so they can store and release a lot of energy there. Kangaroo rats, on the other hand, can't really do that. But yeah, they're pretty cool. Anyway, I'm sorry. So when we talk about reabsorption of the proximal convoluted tubule, okay, so here is where we've got all of this stuff occurring, right? So within the proximal convoluted tubule, we've got our filtrate flow occurring here with all of our vasculature associated with this. We've got active transport, co-transport, and osmosis forcing solute and water out of the filtrate back into the circulatory system. So salt molecules like amino acids, glucose, fructose, trace elements like sodium, potassium, calcium, bicarbonate, and chlorine all get reabsorbed via active transport and co-transport. Things that would be actively transported are going to be like sodium and potassium, potentially chlorine as well. But things like glucose, fructose, amino acids, those sorts of things will most likely be co-transported. So they could be, it'll require a little bit of energy, but if you force like a bicarbonate ion across, chances are you can allow a glucose molecule to go with that. Solutes plus 65% of the filtrate volume get reabsorbed from the proximal convoluted tubule and enter the peritubular capillary and then into the peritubular vessels. Water moves by osmosis from the proximal convoluted tubule into the circulatory system here. Okay. So reabsorption in the loop of Henle, right? So with our descending limb right here, we've got this really interesting bit of reabsorption technique that we call countercurrent exchange. So by that, I mean the fluid flow of the filtrate here, if we look at just the descending limb portion, is going to be moving down, but the fluid flow of the blood is going to be moving the opposite direction. It's going to be moving up, okay? So what that allows for us to do is to kind of create this sort of pseudo concentration gradient that's going to control the movement of things like solutes and water, right? So we do have a little bit of solute diffusion that occurs in the descending limb and also in a small part of the thin portion of the ascending limb over here. But, our big focus is on water. And so when you allow for these fluids to pass by in opposite directions, a couple of things can happen. You create these concentration gradients, and not only can you transfer different kinds of solutes across, because if you've got less water over here and more water over here, less water over here, then that's going to allow for that fluid or that water to move down its own concentration gradient, down its osmotic gradient, right? So that's the controlling mechanism right here. So I'll type that in here as well, countercurrent exchange. Right. So countercurrent exchange is what is the physiological mechanism that's controlling the movement of this fluid here. So the wall of the thin loops of the thin segment of the descending limb of the loop of Henle is permeable to water and to a lesser extent solutes. The interstitial fluid of the medulla of the kidney and the blood and the vasa recta have a high solute concentration, so in other words, less water. Therefore, water moves by osmosis from the tubule into the interstitial fluid and into the vasa recta. Okay. An additional 15% of the filtrate volume is reabsorbed. To a lesser extent, solutes diffuse from the vasa recta and interstitial fluid into the tubule. Okay. Most of our solute reabsorption occurs in the ascending limb, right? The thin segment of the ascending limb of the loop of Henle is not permeable to water, but is permeable to solutes. So we're going to be retaining water, or water is going to stay in the waste product over here. So water can't move across right here. We don't have the necessary proteins. Oh my gosh, what's the name for them? So I don't know if this is the case for us, but it's the case in plants. We have specific proteins that let water move across membrane, or across membranes called aquaporins. They're little pores that only allow water to move across. And if that's the case, which I suspect that it is, there's specific proteins that allow for water to move from a space here across a fluid barrier or across a cell membrane. The reason water can't naturally diffuse across a cell membrane is because of the anatomy of the cell membrane. Water is polar and the membrane is what we call nonpolar. They don't mix. It's like oil and water. You can't get those two things to mix. So water will stay in this area because of that polar-nonpolar association. But if you allow for these specific channels that only let water to go through, water can move across. And that's probably what's happening over here. I haven't confirmed that, but I would imagine that we do have aquaporins in this area, or a protein that does a very similar job to aquaporins. Okay, and so then reabsorption, the thick limb of the ascending loop right here. So remember, water is not allowed to pass across. We don't have the necessary proteins, but we can actively and co-transport, different kinds of solutes across this area, right, to hyperconcentrate this area of the nephron. And so again, what that means is the concentration inside of the collecting duct here, the ascending limb of the loop of Henle, is going to be very dilute. So the milliosmol concentration, remember, once we hit the distal convoluted tubule, gets fairly low. It's around to 100 milliosmoles, which is far less than the natural concentration of the cortex that it ends up at, right? So that's what's going to allow for water to be reabsorbed within the distal convoluted tubule, right? So water moves via osmosis from the distal convoluted tubule and the collecting duct, and then we can have more active and co-transport of other solutes through the collecting duct back into our circulatory system. So what you should be noticing here is that a lot of the physiology of the filtration system is, we're kind of making up for the fact that the glomerulus itself doesn't really select for what gets into the filtrate of the kidney. So it's important that we maintain a lot of safeguards to be able to retain important ions, minerals, nutrients, and things that are normally just being processed as waste, but are important for other bodily functions, right? So there's a lot of kind of cleaning up after the fact of the filtration system within the renal corpuscle in the form of these active transport co-transport proteins, along with osmotic potential controlling the movement of water. Right? Okay, so here we've got a hormone pathway as well. So this is going to cause, it's going to help control blood pressure. So if we end up decreasing blood pressure, one of the hormones that the kidneys can secrete is this hormone renin, or renin, some people call it. So renin is going to get converted, or is going to convert what we call angiotensinogen, which is a hormone released by the liver, and it's going to convert angiotensinogen to angiotensin 1. Angiotensin 1 within the bloodstream is then going to move out to the lungs, where it then gets converted into angiotensin 2. Angiotensin 2 then moves back through the circulatory system into the adrenal cortex, which is going to increase aldosterone. So it's another hormone that's going to control the sodium concentration absorption in our kidneys. So it's going to increase sodium and water reabsorption. So we're going to make sure we retain a bunch of salt and we retain a bunch of water. So if blood pressure drops too much, that hormone renin gets released from the kidneys and is going to help kick off this cascade to increase our blood pressure back up to normal, right? So not only that, but we can also have thirst reflexes trigger as well, right? So an increased blood pressure, or increased blood solute concentration, or a large decrease in blood pressure is going to increase antidiuretic hormone release. So this will cause you to not have to urinate or not have the sensation of urination, right? So the ADH affects blood volume by controlling how much solution gets released with the collecting ducts. So we're going to make sure that we retain as much of that as possible, and that's going to affect the juxtaglomerular cells that are going to control what kind of filtrate gets into the renal corpuscle and what doesn't. So it increases water reabsorption and results in decreased solute concentration and an increased blood pressure. So this isn't necessarily concerned about salt concentration, more just about raw water conservation. Okay. So then we have atrial natriuretic hormone, and this is going to help control blood pressure as well. So the heart can sense an increased blood pressure within the right atrium, which is then going to secrete this atrial natriuretic hormone. So once that hormone gets released into the circulatory system, it's going to have targeted effects on the kidney, which is going to increase sodium excretion and increase water loss. So if blood pressure gets too high, ultimately atrial natriuretic hormone decreases blood pressure. So if there's too much blood pressure that's coming back from the systemic circuit, or it's like, all right, I got to control this. And so it does that through natriuretic hormone. Okay. So a lot of things like blood pressure are controlled by blood volume. And so we've got different kinds of pressure and chemical receptors in the forms of different baroreceptors, chemoreceptors. We have other organs that are going to control the movement of hormones within the circulatory system by either dropping or increasing blood pressure, right? And a lot of that is sensed by those juxtaglomerular cells within the renal corpuscle or adjacent to the renal corpuscle. So those cells can sense whether or not there's a drop or an increase in blood pressure, right? So we've got several different homeostatic pathways right here. So we've got a normal blood volume range, high blood volume, induces elevated blood pressure, right? Which makes sense. If your tubes, if you try and fit more fluid into a tube, you're going to increase the size of that up to a certain point. But if you keep filling that tube up with fluid, you're going to have pressure that's pushing against the wall of that tube. And our body has pressure receptors all over the place that respond to that. So the pituitary gland is going to respond to that, the kidneys with those juxtaglomerular cells, the heart with atrial natriuretic hormone, or ANH, and then blood vessels with baroreceptors just straight out can respond to these kinds of things, right? So we've got effectors with decreased ADH secretion, decreases water reabsorption by the distal convoluted tubules and collecting ducts. So we want to get as much water out as possible. We need to drop blood pressure as much as we can. We want to, so decrease aldosterone and increase atrial natriuretic hormone, decrease sodium reabsorption from the distal convoluted tubule and collecting ducts. So more sodium and water are lost in the urine, which decreases blood volume. So the volume of the filtrate will increase as a response to elevated blood volume, right? And then we've got the opposite effect over here. Okay, so that ultimately brings blood pressure back down. And we have opposite effects if blood pressure drops too low. We're going to secrete antidiuretic hormone, or it's also called vasopressin. We're going to release renin from the kidney. We're going to have the angiotensinogen to angiotensin pathway, which is going to help with aldosterone secretion, which is going to increase salt reabsorption. It's going to increase water reabsorption, and we're going to bring blood pressure back up to normal. Okay, so the ureters, right? So now we've kind of passed from the kidney into the ureter. I think we should be able to finish today, all right? So as I mentioned, the ureters themselves, so we've got, connective tissue that makes up the inner lumen of the vessel right here. We've got smooth muscle and then an outer layer of connective tissue, right? So the inner lumen right here is made up of a lot of transitional epithelia. So that just means that these cells can stretch, right? And so the the bladder itself has a region of non-transitional epithelia that we call the trigon of the bladder. And most of the other cells surrounding it are what we call transitional epithelia. So that can stretch, right? So the reason the trigon of the bladder doesn't stretch is because we have connections from the ureters into the bladder right here. As the bladder stretches, okay, it's gonna block or occlude and pinch off the ureters here. So as fluid is moving into the bladder gets full, and then that's gonna trigger our micturition or our urination reflex. A problem with holding urine for too long is that sometimes you can have urine backflow into the ureter. You can cause kidney infections, you can cause kidney failure in really, really severe circumstances. Or sometimes if you can't urinate, if you have like a bladder or a urethral infection that can prevent you from urinating, this can also cause kidney infections as well. And so, obviously kidney failure is a really big deal because, if we just address the factors that we talked about previously with things like blood pressure, if blood pressure goes through the roof and you can't control that, then you die, right? Arteries, vessels rupture, you have aneurysms all over the place, you end up bleeding out internally, okay? So kidney failure and kidney infection is a very, very serious thing, and there's other things that can, attribute to that. Something I didn't mention earlier was glomerular nephritis, or just nephritis or glomerulitis. Inflammation of the different functional units of the kidney can impair kidney function, right? And so a lot of the time, the ways that you can tell if something is up, it's pretty simple, is that there's blood in the urine, right? If you have blood in the urine, or if urine smells really weird or really bad or foul or sour, that's usually not a good inclination. Another, kind of quick test for things like diabetes are as if urine smells or kind of tastes sweet. But it has to be like, pretty noticeable for there to be any kind of inclination towards diabetes. But kidney infections are another thing that's kind of close to my heart, because last year, my wife and I, we had just moved in, or we'd moved back to Redlands, and we had gotten our first animal together. We had gotten our first cat together, and poor little guy, we only got to have him for a little bit less than a year, but he ended up having a occluded urethra, because sometimes with cats, their urethras, the anatomy of their urethra is such that it gets kind of small and convoluted, and it sometimes can get blocked. And really quickly with cats, what can end up happening is their excretions can be very, very concentrated, and they can form like different kinds of salts or crystals in their bladder that can block the opening of the urethra, and it gets in it. So if this gets blocked, urine backs up in here, can cause infections, and then that can backflow into the ureter and then the kidneys and cause kidney failure. And so, the common nomenclature for that is just called, your cat is blocked. And any of you other cat owners out there, the way that you can look out for this is, checking for UTIs, urinary tract infections. And one of the easiest ways to check if a cat has a UTI is if they start peeing in places that they, don't usually, right? If they pee outside the litter box, or if you keep them outside and they, start peeing inside on things like that, that's an indication that they have a UTI or some form of that. You definitely want to get that treated sooner rather than later, because treatments for these things, I can tell you from personal experience, cost thousands and thousands of dollars, to treat urinary tract infections with animals, especially cats. And so, unfortunately, the poor guy, we did several surgeries on him to help correct the issue, but his urethra just kept getting blocked, and so there wasn't anything that we could do. So, we had to say goodbye to him, but we were very lucky to have him for the year that we did. And so it was, yeah. So, oh my gosh, did he? Maybe the PU surgery, Alex, perineal ureostomy, where they amputate. I don't know if this is what happened to you, but the ultimate thing that they can do is they can actually amputate the penis to make the urethral opening for the animal larger, so that it doesn't get blocked with urinary salts. Yeah, males unfortunately do get that a lot, and it's a horrible thing, but yeah. So, learn from me. Make sure you monitor their health. And it wasn't that we weren't; it was just the poor guy. His just anatomy was against him. So it is what it is. But we're lucky we've got two new little guys now, and they're awesome, and they get to meet their brother here in hopefully not soon, but eventually, right? So, okay, moving right along here. So as I mentioned, the micturition reflex, the sensation of urination, right? Really, in a nutshell, is caused by baroreceptors that are attached to the walls of the bladder that are going to respond to changes in the structure of the transitional epithelia. So as the bladder fills up with urine, all that urine is going to push on the wall of the bladder here, and that's going to send signals back to the brain, right? So the ascending pathways carry an increased frequency of action potentials up the spinal cord to the pons in the cerebrum, where the urinary bladder becomes stretched. This increases the conscious urge to urinate, right? The descending pathway carries action potentials to the sacral region of the spinal cord to tonically inhibit the micturition reflex, preventing automatic urination when the urinary bladder is full. Because normally, if you didn't have this, you would wet yourself, right? And so this is one of the reasons why incontinence is an issue in elderly people, because these neurons tend to degenerate. They tend to go away. And if you can't control that, then you have unconscious urination. And that's part of potty training, is training the brain to recognize these signals and training these muscles to control and not allow for just automatic urination, right? And this is another reason why different kinds of spinal injuries can actually affect the ability to urinate on one's own, because if those neurons get damaged within the sacral region, they control the ability for you to control the sensation for urination. They also control other things in that region as well. So the ability to achieve erections or achieve orgasm are all controlled by neurons that exit the spine in the sacral region. Okay. So it's another reason why the urinary system as well as the reproductive system are closely linked to one another. Okay. So urine in the bladder wall stretches the urinary bladder. Action potentials produced by stretch receptors along the pelvic nerves. The sacral region, action potentials are carried by parasympathetic nerves to contract the smooth muscles of the urinary bladder. It's going to force urine out through the urethra and allow the external urethral sphincter muscles to relax to allow a constant flow. Okay, so here we have total body water percentages, approximate body volume. So total body water, 75 for an infant, males 60, adult females 50 intracellular fluid here in plasma, right? So we've got those values here. Let's see. Yeah, so really just, our brain can kind of trick us into having a sensation for thirst as well in response to things like increased blood concentration or a large decrease in blood pressure. So if our blood becomes too high in concentration, that is an indicator that the blood volume is low, which means that we don't have enough water in our blood. So that's going to trigger a thirst response to allow us to ingest water or take in water. Okay. And there's some interesting psychology behind people who are severely dehydrated and whether or not they will drink water that's been poisoned or not poisoned. And there's I want to say there's an actual threshold of dehydration where people will forego the reflex of drinking contaminated water if they're dehydrated enough. Okay. Potassium regulation as well is important. So if blood potassium levels increase, okay, so increased blood levels of potassium act on the control center in the adrenal cortex to increase aldosterone secretion, bring blood pressure back down, increase blood pressure. So the increased aldosterone increases the rate of potassium secretion from the effectors, distal convoluted tubules, and collecting the acid of the kidney into the urine. And then blood potassium levels go back to normal. Okay. Same thing with pH. So the pH is going to be controlled by that renin-angiotensinogen-angiotensin conversion that occurs with the lungs, right? So the kidneys get linked to that. In the lungs, we can control our breathing patterns to if our pH decreases, then we have increase the depth of respiration, which decreases blood carbon dioxide. So we're going to increase our rate of breathing if our pH increases, if we're undergoing acidosis, to get our blood pH buffers back into action right here. And then we do the opposite of that, where we can slow our breathing rate to increase the amount of carbon dioxide in our blood, which is going to lower the blood pH back to normal operating ranges around 7.4. Okay. And then finally, issues with the kidneys, as I had mentioned. So glomerulonephritis, acute glomerulonephritis, and chronic glomerulonephritis. So again, just inflammations and infections of each of these areas for either short periods of times or long periods of times. And then ultimately kidney failure, so acute and then chronic failure, right? Things like kidney stones can form based on diet or salt concentration. Kidney stones, or if any of you guys watch Friends, kidney stones, right? Yeah, so, wow. All right, guys, we finished. Okay, so this is it for chapter 18. So it's nice because then we'll get a lot of time to address or talk about reproduction. Okay, so that'll be good. All right. Do you guys have questions about anything? I guess really quick, for the Thursday folks, you guys don't need to stay in this for lab tonight. It's posted on YouTube, so it's posted on the YouTube channel. You guys can go and watch the lab lecture there. All right, it's all about the digestive system. Okay, so that's it, you guys. If you have any questions, I'll stay around for a little bit, but if you are good to go, have a good evening.